I'm going to give an overview of my research work in the last 5 years, which is focused on the regulation of cellular processes by phosphorylation cycles, and in particular by multi-site phosphorylation. The communication of the cells with their environment is important for the regulation of cellular activities, and phosphorylation cycles play an important role in the transmission of this information from the cells, from the cell membrane into the nucleus. This information is transmitted by a very complex network of signaling proteins which are activated by phosphorylation. The addition of a phosphate group to a molecule can activate the protein, and in the presence of a protein kinase, and when the stimulus is not more there than the phosphate group is removed by the action of a phosphatase. Phosphorylation is the most important way to regulate the protein function, and about 30% of the proteins in the human proteins are phosphorylated. When the level of protein phosphorylation is not correctly regulated, and it can induce several diseases, for example, cancer. Several kinds of proteins are regulated by phosphorylation, for example, enzyme kinase, cell receptors, transcription factors, and enzyme inhibitors. And phosphorylation can control the protein function at several levels. For example, the binding of other proteins, they can induce conformational change, and regulate also the subcellular localization of a protein, the binding to DNA, and the transcriptional activity. When some proteins are phosphorylated, then can be degraded, and then phosphorylation can also regulate the stability of the protein. The mathematical modeling is very important to understand how different factors regulate the phosphorylation of the protein, and how the phosphorylation regulate different cellular processes, the cellular outcomes. And one important point is that many of these processes are non-linear. For example, the formation of protein complexes, there are feedback mechanisms and crosstalks between signaling pathways. And this can induce the systems properties like the threshold for the activation of a protein, or a bistable response, or the oscillation in the protein concentration. Most of the proteins are regulated at more than one residue, and this can create a large number of phosphorylation states and protein complexes. For example, the EGF receptor is phosphorylated at 9 sites, and that means that there are 512 possible phosphorylation states. One challenge, one problem in the app is that the application of traditional enzyme kinetics in signal transduction is not always possible, because this is based on the assumption that the enzyme concentration is much lower than the substrate concentration, and this is not true in signal transduction in many cases. For example, in signaling MAP kinase cascades. Try to investigate in a systematic way how different thermodynamics and kinetic factors affect the phosphorylation of the protein, the kinetics of phosphorylation, and the dose response. How the phosphorylation of level of the protein depends on the kinase activity. And we found that there are two key parameters that determine the response, the protein phosphorylation. One is the degree of enzyme saturation, and the second is how the product of the phosphorylation, the phosphorylative substrate, can inhibit the phosphorylation reaction. And I show here in this phase diagram how when these parameters are changed, then the complex response are generated even in the case of a single phosphorylation cycle. Here, when the enzymes are saturated by the substrate and there is no, the phosphorylation is not inhibited by the product, then there is a switch-like response, and also in the case when the enzymes are not saturated and the phosphorylation is inhibited by the product. And other response are the great hyperbolic response and also a dual threshold. When proteins are phosphorylated and more than one residue, other factors should be considered. And one is in which order are the phosphorylation sites are modified. And it can, there are two basic mechanisms. One is that the phosphates are in the sequential order phosphorylated, and the second is in the random order. And when the phosphorylation is random, there is a large number of phosphorylation states and pathways. And we analyze the consequence of these two mechanisms for the protein phosphorylation. That when the proteins are randomly phosphorylated, they require higher kinase activity, and but the kinetics is faster. And when the proteins are phosphorylated in a sequential way, then there is a threshold for the activation of the protein, and the phosphorylation is slow, in particular when the kinase and phosphatase activity are balanced. And this is important because depending on the processes that should be regulated, in some cases, for example, in the cell cycles, many proteins are randomly phosphorylated because these processes should be fast. And in other cases, it's more important to have a good threshold for the activation. We investigated the role of multi-site phosphorylation in two cases. One case is the activation of the transcription factor MFAT. MFAT is a central regulator of gene expression in the immune system, and is a major target of immunosuppressive therapies in organ transplantation. The activation of MFAT is regulated by its cellular localization. When MFAT is in the nucleus, activates gene transcription, and the subcellular localization of MFAT is regulated by its phosphorylation states. When the cells are stimulated, then the calcium concentration, intracellular calcium concentration increases, and MFAT is dephosphorylated by the calcium-dependent phosphatase calcineurin. And when and then MFAT is imported into the nucleus. When the signal decays and the calcium concentration is low, MFAT becomes rephosphorylated by several kinases and is exported to the nucleus. Several phosphorylation sites are involved in the MFAT activation in the mechanism, and they are situated in two main domains, a serine-rich region and a serine-prolin region, and are phosphorylated by different kinases. And one question is how these phosphorylation sites cooperate in the activation of MFAT. On experimental data, we develop a mathematical model that can explain MFAT activation by a conformational switch. That according to this model, MFAT can exist in two different conformations. One active conformation where the nuclear impulse signal is exposed, and an inactive conformation where the nuclear impulse signal is masked. And the probability to find MFAT in one of these conformations depends on the phosphorylation state. When MFAT is dephosphorylated, the active form has a higher probability, and each phosphorylation progressively increases the probability of the inactive conformation.